We're very excited today to welcome Dr. Ernesto Nakayama and Dr. Lisa Bramer coming to us from the PNNL, or the Pacific Northwest National Laboratory. Tom Metz is also a big part of this work, but he's not available today. However, he was originally going to speak regarding this topic, and he is part of the work. So today's talk is plasma protein biomarkers predict the development of persistent autoantibodies and type 1 diabetes 6 months prior to the onset of autoimmunity, coming from the TEDdy study. Just a little bit about our speakers. Dr. Nakayama received his degree in biological sciences from University of Sao Paulo in Brazil and a PhD in pathobiology from UTexas El Paso. After postdoctoral training in Purdue, he returned to the Pacific Northwest National Laboratory in 2015, where he's currently a senior scientist and the biostructure and function team leader. His research has been focused on the application of advanced mass spectrometry technology, such as proteomics, lipidomics, metabolomics, and analysis of protein post-translational modifications to study complex biological systems. In diabetes, Ernesto has been studying biomarkers predictive of type 1 diabetes development, molecular mapping of the pancreas, and mechanisms of beta cell dysfunction and death. Dr. Lisa Bramer received her bachelor's degree in mathematics and psychology from Purdue, and then earned an MS and PhD in statistics from Iowa State University. She joined the PNNL in 2013 and now is a senior data scientist. Her research focuses on the development and application of statistical machine learning and data integration methods and models for mass spec-based omics data. She's been involved in the identification of biomarkers predictive of type 1 diabetes development, in addition to other human health and disease research areas. And we welcome you both, and we really look forward to the talk.
All right. Oh, thank you, Monique, for so kind an introduction. And it's my pleasure to be here today. So I'll get started, and Lisa will take over more the statistical part.
Great.
So a little bit about type 1 diabetes. I think most of people here know what it is. So it's autoimmune disease. So you have like the death of the beta cells of the pancreas. So by autoimmune response or inflammation. It has a genetic predisposition from these HLA alleles, and there is also a phase that you have these autoantibody that appears before the onset of the disease. There is a possible link with viral infection, but I don't think it's very consolidated in the literature yet. Although I think there's some very strong evidence. So a little bit about the process. You have this autoimmune response. I don't want people to try to make sense out of this. It's just the thing that you want people to remember here is that in this process you have a lot of these cytokines being released. So and also you have all these cells being recruited at the site. That really drives the beta cell to die. So a little bit about the disease. The treatment is based already insulin nowadays. So you can have other drugs that can also help with some of the other symptoms. There is no cure or remission for type 1 diabetes. And diabetes complication can reduce life expectancy by about 12 years. So it's very significant. And the beta cells don't regenerate, so you cannot just regrow them. And so for us to be able to prevent this disease to ever happen is like an ideal scenario. So the goal in our approach, so our goal is to identify and validate biomarkers that can predict the onset of the disease. And our approach is to do global and target proteomics. And then here I'll turn to Lisa to talk about a little bit about the study design and the statistical part.
Thanks, Ernesto. So we approached this study with two different phases. An initial phase where we were doing discovery, and in that case, we are looking for potential targets downstream where we've got our candidates, and then we moved in from there after doing statistical and machine learning, moved into a validation phase. And if you want to go to the next slide, Ernesto.
Yeah.
So in the discovery phase, we had about 2,200 samples. What we did here is we actually pooled several time points from individuals in order to run samples. So pooling time points prior to things like seroconversion or after seroconversion. And then in the validation phase, we actually ran every person's individual time points through, which amounted to running about 6,400 samples. You can see here how many QC samples in the instrument time that took, as well as if you're interested in some of the quality control tools that we used. We have a publication out about QC art, which details how we were able to identify lower quality runs on the fly off of the instrument to ensure that samples were run as we went and we got the most of our data.
Lisa, if you or Ernesto can drop that paper link in the chat, that would be great.
Sure. Can you go to the next slide, Ernesto?
Yes.
Thank you. So yeah, so I mentioned in the discovery and validation phases, the designs were a little bit different because we were doing untargeted analyses in the discovery phase. So we have 4 different groups. One group where autoimmunity is the endpoint of interest, and one group where type 1 diabetes is the endpoint of interest, and then matched case control pairs for each of those conditions. TEDdy does have individual time points and samples from each of those time points, but because we're doing untargeted, in order to run samples more quickly, what we did is, you can see here in the orange, plasma samples that were before seroconversion were pooled together and then matched up to the case control pairs, or the controls, and time points after seroconversion were pooled together. In the validation phase, you can see the difference where each individual time point was run. Here, we're, instead of untargeted proteomics, we're running targeted proteomics, and we're comparing, or we have a control at each time point. And I think, Ernesto, there's a question in the chat about pooling. I don't know if you want to address now or how to make pooling samples same volume or same amount of proteins.
Yeah, it's by volume, because we had also to deplete the plasma to get better coverage, so we couldn't measure that beforehand. So we just pooled by same volume.
And then if you want to go to the next slide, Ernesto. So this is kind of the general pipeline that we took. Of course, we had experimental planning where we did power analyses, selected the cohort, and we were blinded. Samples were collected by TEDdy and underwent immunodepletion and protein digestion, and then we collected the data using standard untargeted proteomics instrumentation that Ernesto can go to in more detail if that's of interest. All the way through with the quality control, the peptide identification, and then conducting statistical analyses where we did several comparisons of interest, but primarily in that discovery phase, the comparisons of interest were for the group that has autoimmunity as an endpoint, asking if before seroconversion, what are the differences between that cohort and the control, and for the endpoint with type 1 diabetes, again, before seroconversion, what are the differences compared to the control group? You can go to the next slide, Ernesto. And once we have that identified list of peptides, in the discovery phase, we measured 36,252 peptides that mapped to 1,720 proteins. Some standard filters and quality control pre-processing steps that we tend to take. So we ensured that in order for a peptide to be considered, it had to be observed in at least 2 samples per group. So that's either the autoimmunity or the type 1 diabetes endpoint or the control group. And we really focused on proteins that have at least 1 peptide or at least 2 peptides that map to it. That filtered the data set some down. Then we also looked at the variability inherent to each peptide and protein and removed things that were highly variable. Where you could see we resulted in about 2,900 peptides that mapped to 1,200 proteins. And that is where we did protein statistics for 4 different comparisons. At this point in the study, we were still, 6 different comparisons. At this point in the study, we're still blinded. But those comparisons essentially boil down to if the autoimmunity endpoint was different from the control, either pre or post seroconversion, the same for the diabetes, and then also looking at if there were differences between the time change. So were the changes with time, over time, the same or different in the subjects that have the autoimmune endpoint versus the diabetic endpoint? After those 6 statistical comparisons and false discovery rate adjustments, we ended up with 376 significant proteins that we then passed off and said these are the proteins of the largest interest to us.
Okay, thank you.
So just a quick question in the chat from Dr. Hayun Kim. How did you just to clarify, how did you make the pooling samples? Did you use the same volume or the same amount of protein?
It was the same volume.
Okay, thanks for clarifying.
Yeah, so here is like a heat map of some of the pathways we see enriching in these proteins that were significant change. So the comparison here, just to remind everybody, is that this I1 is just for the autoimmune, the people that got autoimmunity before the seroconversion. I2 is after, I3 is before versus after, and the T is the same time point, but to people that develop type 1 diabetes. But one thing that is noticeable is that they don't have exact like the same proteins. You see that the extracellular matrix is significant in all of them, but the proteins are quite different, you know. And one thing that also is very distinguishable, I think, is to a certain extent is expected is the antigen presentation. You can see more in the T1 and the T2 conditions to be significant. And sorry that I forgot to mention these star ones are significant ones, but not as much on the I comparisons, you know. So from there, we look at these more in different pathways, but we can divide these into a few of them. Actually, like antigen presentation is one of those. Complement and coagulation, then extracellular matrix, inflammatory response, and metabolic proteins. You know, I think some of these metabolic proteins, people might think that is just because, you know, you already start to have some dysfunction of your insulin, but some of these comes from much earlier time points. You know, so we think also that there might be something there too. You know, we compare our data set with other in the literature, so we perform a systematic review of the literature. So what it allows us to do is now to see which proteins are being shown over and over by several studies, and we can see things like CO3 is complement protein 3, C3, and C4A, kininogen, for example, clustering. Those are things that has been coming as a candidate over and over again. So I mean, so for future studies, that's something that we should use for prioritizing. So just a like kind of summary that we had in mind from what we see in the pathways is that we have the beta cells, and you see that there is some signal that for like the phagocytosis and then the antigen presentation to lymphocyte, and also signals from the cytokine production, and also the inflammatory signaling and the metabolism here. So move forward. So these, Ethan, you want to take this over now?
Sure. So once we moved into the validation phase method, again, we're in a scenario of being blinded.
Sorry.
No, you're fine. Right, very similar process to before, but doing looking at targeted assays from those selected potential biomarkers, all the way through to collecting the data and doing alignment and intensity extraction, then moving into quality control and doing statistical analysis and multiple test corrections to do the biomarker validation.
Thank you. And then these, because we had like these 376 proteins, or ideally we wouldn't want to measure all those in the target proteomics, but the technology doesn't allow to get that many proteins, because we go for as many 5 peptides, when we start per protein. So that would be like about 2,000 peptides, which the system wouldn't allow. So we had to do some down selection. So we did some p-value correction, and then also we look at for some other things, like if the peptide is be able to detect in several samples. So it was another criteria. So we also did machine learning to see which ones are very good for predict the outcomes. And also we consider things that were had overlap with the literature. So in the end, we got 167 proteins, and we target those for the validation phase. And from those, we design over 800 peptides, and we try to measure most we could. And I think we got about 600 peptides that we can we could measure. And Lisa, do you want to go now for this, and then the AI part?
Yeah. So we did do some of the standard comparisons that was mentioned in the discovery phase, but what the validation phase really allowed us to do is we now had individual time points at different times prior to seroconversion. And so, for example, we could look at the subjects that had autoimmunity as their input, and we could take where they were 3 months prior to the visit where they were diagnosed as having autoimmune markers, and run machine learning to ask, are there any biomarkers here that are predicting those subjects who will become autoimmune versus those who are controls. And the same thing for the diabetics, right? Where diabetes is the end outcome, asking what are the differences that a machine learning model would let us use at 9 months prior, 6 months prior, 3 months prior, or at the time of autoimmunity. So what you're seeing here are some of the markers that were identified based on using machine learning models. This would be logistic regression with a elastic net penalty on it to do variable selection. And what we found is that we got actually very good predictive accuracy. I think, Ernesto, if you go to the next slide. So on the left is the outcome of autoimmunity as the endpoint. On the right is the type 1 diabetes. And this is 6 months prior to seroconversion. You can see the receiver operational characteristic curve where we are doing prediction of those subjects versus their controls and getting AUCs between 0.87 and 0.91 in a cross-validation scenario. So we're able to very accurately predict who is going to convert 6 months prior, comparing those cases to those controls, and then identifying which potential peptides are the predictive biomarkers driving these predictions.
So before we move there, also Lisa looked at 3 months and 9 months before, and the results were quite similar too. So I think some of the signatures might be stable enough that we might be able to get a good biomarker out of it. So the summary about this part of the study is that we identified 376 proteins that were regulated in the different comparisons. We found an enrichment pathways such as antigen presentation and extracellular matrix among the regulated proteins. And then we cross-validate just by regular statistics, 83 of the 167 targets for the validation phase. And then we have this multi-panel that can predict both T1D autoimmune onset. Mark?
Ernesto, yeah, I was just going to say Mark Atkinson has his hand up for a question.
Oh, geez. Again, this is a fascinating presentation. Great job to both of you for presenting it and for Monica to bring it forward. Can you go back one slide? I'm sorry, I was slow to figure out how to hit the So again, congratulations. What I'm wondering in here is if you look at this, do you believe, and then I'm looking at the entirety of your presentation, and maybe you were going to get to this and I'm just too early. Do you believe that the best markers of this process are going to be immunological markers, beta cell markers or something, or the two in combination? And how do they correlate with each other?
It's actually a combination. We have, from the panel, we have things that are more immunological, some things that are more like things like metabolic proteins, like lipoproteins, so it's a combination panel.
So can you draw, rather than descriptive, can you draw some sort of projected mechanistic notions from this? Like chicken versus egg, which comes first, or is one driving the other? And again, I know it's speculative.
I think, so that's what it's my feeling, Mark. I think there is some like a metabolic predisposition to not just the immunological predisposition we know about the HLA, but these kids, they have something, the metabolisms are different, and they are from like most of the time they're born. So if you see there is the OSIRIC group just published a paper on showing that the bioacid is actually different. And that is from very young age, before a year old. The bioacids are very different from the group that develops type 1 diabetes, compared to the control. So there is something, and the bioacid is something that's important for you to absorb things like fatty acids, So I think there is something to the metabolism too, that's not just like the immune response that really drives the beta cell to die,
So I don't want to hog all your time and being respectful for others, but if you can go back to like 5 slides-ish where you had your pathways. Keep going. One more, maybe. Yeah, here.
Yeah.
So, nope, back down one.
Yeah.
So how do you what's the latest systems approach to taking these maps in trying to put them into some sort of time frame. Is that possible these days, or is this kind of just where we're at, which isn't a bad thing?
No, I think it is possible. It's something that we're trying to address. Lisa and I are writing a couple proposals for this NIDDK call to use the TEDdy samples. So one is focused on the protein biomarkers, and then another one that we're writing actually is based on the fatty acids, because we see some very interesting signatures of fatty acids as well. To be honest, like, it's a little bit hard to really delimitate, because a lot of this data that is available is just a single time point. So as we're gonna collect more these systematic studies that go longitudinal samples, then I think we're gonna start to understand like where each process starts. So if they start from very early age, or say some that maybe just starts with the autoimmune response, and then we're going to start to have more definition, like in terms of time, evolution.
Because again, I think, and again, we can all look at this. It's like art. Like you look from the left, you look from the right, you look from the center, and you see different things. As I look at this, I'm saying, is it the metabolic proteins at the right-hand side driving the left, or is it at the level of the ECM or cytoskeleton, or is it on the immune side? And how are these things going? But again, fascinating presentation. Congratulations to both of you. I think this is huge. Great.
Yeah. So let me move forward. And the last few minutes, I want to focus on some new directions that we are moving now for these biomarker studies, which is the extracellular vesicles. So extracellular vesicles, EVs, they're considered excellent source for biomarkers, but there is a couple problems. There are a couple problems. One is that it's very hard to get rid of the contaminants, and the second one is that it requires really large volumes of sample. So they are the 1 to 2 mL of plasma, which is a lot. So our goal, and so the first part, we're trying to identify what is the specific EV protein, and to do this, we did this meta-analysis of EV proteomics data set. So the concept here is say that if the hard part is to get rid of the contaminants, we need a lot of samples. Maybe that if we know what the contaminant is, we can just really just measure everything together and really just say, oh, this is a EV protein, this is contaminant. In the concept we got is that the different methods to purify EVs, they're going to have different proportions between contaminants. So if you see method 1, 2, and 3, they have different proportions between contaminants and EV proteins. So we got this, and it's kind of hard to get all this data in a single lab. So we got all the data that's deposited in these repositories, and they come from different types of methods for purification. For example, the size exclusion chromatography, centrifugation, ultracentrifugation. So this is cushion centrifugation. This is gradient ultracentrifugation. PP is a polymer precipitation, and Prosper is thermal precipitation with organic solvent. So then we process all this data in the same way, and then we compare across them. So our approach is say that, so if they have different proportions, so then we can cluster them based on their abundance, so then things that are in vesicles should have similar profile than vesicle markers, and things that are contaminants, they should have profiles that are more similar to contaminants, and that's what we got here. And then to validate this, we did some enrichment analysis, which we say where the clusters are enriched with vesicle proteins, and we did a systematic review of the literature to find exactly what are really validated EV proteins by immunogold electron microscopy. And then with that, we found that the cluster 10 and 11 are enriched in EV proteins, And that actually has a true positive rate of over 70%. So we were quite happy with that. And then as you can see here, so serum albumin and apolipoproteins, there are two well-described contaminants, and they are basically depleted from this cluster 10 and 11. So when we move forward to look at the functions now, and then functions make a lot more sense instead like, oh, these, some people would believe that EV was a mini cell, which, after working for 20 years with EVs, it was hard for me to believe, but now it makes more sense because if you look well where they enrich on, so there are things about like cell-to-cell communication, intercellular signaling, lipid signaling, metabolism. So things that are more basic cellular functions like DNA replication or protein translation, they are actually outside of this cluster. So for us, it indicates that there may be those that are not really secret inside the vesicles. So then we try to get back to these biomarker candidates. So we got these proteins enriched in these two clusters that are enriched with the plasma, in EV proteins, and we compared to EV from cells, not from cells, from islet cultures. And also we look at the biomarker candidates we had from Teddy, Daisy in the literature. And we got these 19 proteins. So the 19 proteins here, for example, we were really interested in this PPBP because Tom Metz had a paper before that he validated these as potential biomarkers of T1D onset. So, and also then we went back to look at the PPBP is a chemokine, actually also called CXCL7. And if you look at this chemokine pathway in the plasma EVs, actually you get almost the completed pathway inside the EVs. So all these green proteins were things that we found to be enriched in those clusters that were enriched in EV proteins. So you can see that you can basically have the whole signal transduction there.
Ernesto, we just had a question in the chat from Leandro Balzano. In your opinion, what would be the role of ROS in terms of proteins expression?
It's a good question. I think that there is a role too, so that would cause things like stress, like for example, ER stress probably is linked to that, that would repress the translation of proteins, because ER stress regulates translation. So I think the ROS has a major function actually. Okay, so going back because of this PPBP, where we wanted to really see if we can validate these as a vesicle protein. And we used this technology here. It's called NanoView. So basically what the NanoView is these microchips that, you have, they are conjugated with antibodies against EV markers. And so the CD81, 63, and 9. And there is isotype control here. And basically what it does, it captures these EVs into these chips here. And then the proteins that you're interested in measuring, you just use another antibody with PRO4 to detect them. So when you have the result, you see something like this. So this is a control that we label with CD81. So most of the vesicles should be marked with CD81. But we can see that a small population of the vesicles actually contain PPBP. So you can hear one example here, but you can see they're all spread out here. We then treat mean-6 beta cells and the macrophage with PPBP and cytokines. And actually, in mean-6 cells, the PPBP enhances apoptosis, and in contrary, in macrophage it decreases apoptosis mediated by the cytokine. So we think there is might have some role also in the process of type 1 diabetes development, but that still needs some more studies. So going back to the comparison from the different methods to enrich for vesicles. So here is like the amount of the non-contaminant serum albumin or apolipoprotein on this different methods to enrich. And then if you look at also the enrichment on the EV proteins, there are two methods that are kind of stands out. So we were interested in this size exclusion chromatography because that would allow us to do very fast and automatable and use a very little amount of proteins to analyze the vesicles. So we got this, and now we run this on an instrument called FPLC with this size exclusion column, and this is like only 50 microliters of plasma. So compared to the 1 to 2 mLs that people used to use, now we can go down to 50 microliters. That it's more, I say, I think if people have like their repository of plasma, they don't want like to give 2 mLs because then it would prevent other studies to be done. But 50 microliters, I think it's a feasible amount to be able to, in some of these studies. And here is just a control plasma, but you can see how reproducible the chromatography is. That it just seems to be one sample, but actually the 3 samples are that we've run that's on top of each other. So it's almost, basically perfect reproducibility. We test this set with a small set of samples that we have in the lab, and this was a wildland firefighters that went through a 45-minute simulation exercise, and then we can see that the EVs are enriching this fraction 3 here, and then when we run those in the mass spec, we can find about 800 proteins and 261 proteins that we were previously found to be from EVs. So we're quite happy with the data now, and actually when we look at this more carefully, so the proteins were higher before exercise, were more enriched in lipoproteins. And then the ones that are higher after exercise are actually enriching EVs. We try still to make sense out of the data. We just got this week, but we think it's quite exciting that there might be some function for the EVs because also they are related to things like tissue regeneration. Maybe that's like something that will help like recover after the exercise. So the summary of the EV studies that we use this meta-analysis to refine the composition, the plasma EV. The EVs that we found, they were enriched in signaling communication proteins, and then we found this PPBP as a possible biomarker, not only possible biomarker, but something that might have a direct function in T1D development. And also now we are gonna move to our next stage, and then we already have samples here in-house from Amy Sims from IU that she sent us 20 individuals that are autoantibody positive in 20 control samples that we're gonna run this platform in this platform. And here I'd like to thank everybody that is involved in this study. It's a massive study, 9 years just for Teddy, and we couldn't do without all these wonderful people, they really work very hard in the lab to get this done, And thank you for your attention too.
Thank you, Ernesto. And Lisa, that was a beautiful presentation, really in the frontier of, examining data and, going back into data, going back into Teddy and pulling out new observations, which we're big fans of. And I wanted to open it up to any other questions. Feel free to.
There is one question here in the chat.
Yes.
It's about how many single autoantibody, and that is a good question. Do you remember that, Lisa?
Yeah, this question is from Alexis Karmer. Many of the subjects in both discovery and validation were single autoantibody positive. Did you do protein analysis on any subjects that had reversal of seroconversion during the study period?
I don't think we had any that had a reversal. So I think those were excluded. And to be honest, I don't know about the number of antibodies.
They all tested positive for at least two consecutive visits.
Yeah,
So,
but is that single or multiple? I think that we didn't get the information, but Alex, if if you're interested, that you can write proposals to Teddy to investigate those kind of things, because we as much we want, we cannot we couldn't go and test every single phenotype, so Teddy has a lot of data. So you also can get data for things like lipidomics and genetics as well, and you can make all kind of different correlations. You just need to write a manuscript proposal to them and ask for the data. And
Ernesto,
then,
did you find that process to be relatively easy and timely, writing to Teddy to get access to the data?
It's quite straightforward, but they are kind of very busy, so it might take like a couple months to get back.
Okay, good. And we can see here the Teddy DCC, Jeff Krishner, Kendra Vahik, Steve Fisk, Dina Tui, and Susan Smith, all very involved with Teddy.
Yeah. Mark?
Yeah, so here's my question, and this is more of a hypothetical one. And again, I've been praising you and the team here at PNNL on this. So congratulations, Mike. Question is that in the 1970s, we had autoantibodies and HLA as being markers for type 1 diabetes, and they certainly got better in the 80s and then in the 90s in terms of specific autoantigen identification. And where are we in 2023? We measure genetic risk and autoantibodies. This despite, measures of growth, the IGF-1, IGF-2, trypsinogen, the amylase, endocrine markers, the prohormones, the proinsulin, insulin ratios, C-peptide, methylated insulins. I mean, we have a bazillion candidates, but they never break through in terms of day to day in, day out, markers. What do we do today for Teddy and other things? Autoantibodies and HLA. So my question to you is what do you think? And it's just an opinion question. I don't know the answer myself. What is it going to take to see a breakthrough in order to see non-conventional biomarkers come into areas of diagnosis and prediction, et cetera?
Yeah, I think that this machine learning that Lisa does has quite potential too, because that it's not restricted to just a single. Because when we want to do a biomarker, we usually want to have something that it spikes out and it stays constant for a long time, and then those are like kind of hard, because see that a lot of these processes that we saw are either metabolic or signaling, so they go up and down with time, right? So I think I have a less hope to just be able to tell you that we can focus on one protein and say like this is going to be our biomarker. So I think it will be more like in my opinion, would be something like a multi panels, so then we can stratify multi panels and really go and actually understand where they are in the disease development process too,
Okay, I appreciate that answer. I guess, and maybe today's not the day to answer this call, but I do think that programs like Teddy and TrialNet and the GPAD and Enodia, we've spent 3 decades trying to identify additional markers, but just day in and day out, they're not utilized. And I don't know if we have standards of what it's going to take for these things to become. And then look at Alberto is on the call here. microRNAs, I'll add that to the list. What is it going to take to see additional markers become in the question? And maybe you're right. Maybe it's going to take an AI or some sort of non-conventional way to see these things included.
So the other thing, Mark, I think, in my opinion, if we know a little bit more about the mechanism, like some of these, we just say, oh, these are different. But if we already know, we get to know more about the mechanism, I think people will be more, we will ease people's mind to use them. Because if we just say, oh, this go up or this go down, but why? If we say, oh, yeah, fatty acids say go up because these because these change in metabolism before, or it helps the beta cells to die or something like that, then people will be more willing to use, I think, too.
If I can comment,
Yes.
those are very important questions. I mean, they're very general to the field of biomarker discovery and validation. One of the problems that we have when we look at this, whether it's proteins, microRNAs, whatever, is we have no idea where they're coming from, right? And so pairing these analyses with a couple of things. One is perhaps experimental studies that can at least inform us of whether cells of interest in the pathogenesis of disease would have those proteins, which is probably likely, but then pairing that with a stimulation that perhaps can be done in the living subject to stimulate a response from the cells that are important in the disease pathogenesis. And I think that the metabolic testing that is routinely done is actually a great test because it will stimulate beta cell function. And so if you can measure changes during a stimulated insulin secretion test, right, then you could be more likely to observe changes that are actually reflective of a beta cell response. And so we're trying to implement that. It's not easy to do, but I think that could help a lot.
Yes. One thing that I just came in my mind, I have one slide here, is that for the extracellular vesicles, actually, like these extracellular vesicles from plasma, they come from different tissues, and including pancreas, So maybe this might be something that we can find signatures come direct from the beta cells, But of course, the technology is not there yet. But something like this, none of you might be able to, will be able to maybe capture some EVs that are specific from say pancreas, and then really assess like some specific biomarkers from there, and if they're going up or down,
Yeah, no, these are all excellent points, and thank you for sort of some of the top leaders in the field for weighing in with their impressions and questions. I also would just offer that maybe it would be great to have some input from physicians who interface with patients at diagnosis to capture these time points if it was easier, if there was a protocol in place, maybe coming out of ADA or pediatric standards that, just as if we just want to kind of hark back to the SCID situation, which is different, but there can be some lessons learned. Parents, really lobbied hard to get that SCID, the TREC test implemented in the newborn heel stick. And perhaps there's another place for JDRF or other organizations to lobby hard to get some of this testing implemented in the regular pediatric visits, or even newborn visits, to kind of get at what you're talking about, is the fact that there's possibly metabolic predisposition, fatty acid processing that's messed up from the beginning. So I think that there's a lot of room for improvement, and your work is beautiful. Lisa and Ernesto and Tom, thank you so much for joining us. I would encourage people to reach out with any questions to you directly. And as you said, if people have questions that they think that might be answered by going back into the TEDdy data and running some of these AI and bioinformatics, processes and programs, reach out to TEDdy and try it.
Yeah. And then, if you guys, just, email us, if you have questions or, want to collaborate too. Our email is here at PNNL is very easy. It's our first name dot last name at pnnl.gov. So you now know everybody's email here at PNNL.
Fantastic. Thank you again, and I hope you have a great rest of your day. Thanks
Oh,
for
thank
all
you
the
so much. It
great
was
work.
a lot of fun here.
Yeah,
Yeah,
thank you.
fantastic.
Right. Okay, bye.
Bye.
Bye-bye now.
Bye.